How do clinical trial endpoints relate to statistical power? Imagine trying to find out if a new medicine really works. You need a way to measure success, and that's where clinical trial endpoints come in. These are the specific results researchers look at to see if the treatment makes a difference. Some endpoints are direct, like whether a patient lives longer or feels better. Others are surrogate, like tumor shrinkage, which can suggest the treatment is working even if it's not a final outcome. Picking the right endpoint is a big part of designing a trial because it shows what success looks like. Now, statistical power is about how likely a trial is to detect a real effect if it exists. Think of it as the trial's ability to catch a true benefit. If the power is high, the chance of missing a real effect is low. Power depends on several things, including how big the treatment effect is expected to be, how many people are in the study, and how much the data varies. But one of the most important factors is the type of endpoint chosen. The kind of endpoint affects how much data you need to see a clear result. For example, if the endpoint is overall survival, meaning how long patients live after treatment, you might need many patients and a long follow-up period. That's because deaths happen less often and take longer to observe. On the other hand, surrogate endpoints like tumor response can happen sooner and more often, so trials can reach conclusions faster with fewer participants. This means the statistical power can be higher for these types of endpoints because there are more events happening in a shorter time. When endpoints are based on events, like disease progression or relapse, the number of events observed is what really matters for power. More events mean more information, which makes it easier to see if the treatment is making a difference. If a trial has too few events, it might not be able to detect a real effect, even if one exists. That's why event-driven endpoints are so important. They directly influence the trial's ability to find meaningful results. Sometimes, trials include multiple primary endpoints to look at different benefits of a treatment. While this can give a broader picture, it also makes the statistical analysis more complicated. Testing several endpoints can reduce the power for each one unless adjustments are made to account for this. So, careful planning is needed to balance the chance of finding real effects with the risk of false positives. Choosing the right endpoint is also about balancing clinical relevance and practicality. For patients, an endpoint that occurs quickly or frequently can mean faster results, but it must still reflect a real benefit. For example, a surrogate endpoint like tumor shrinkage can be measured sooner, allowing quicker decisions about a treatment's potential. But the results should still be meaningful for patient health. In real life, when patients look into trials, understanding how the endpoints are chosen helps them see how soon and how reliably results might come. Trials using endpoints that happen more often or sooner can give answers faster. But those results need to be interpreted carefully to see if they truly reflected benefits that matter to patients. Trials using longer-term outcomes like survival provide direct proof of benefit but may take more time and more participants to reach solid conclusions. To sum up, clinical trial endpoints define what outcomes are measured, and these choices directly influence the statistical power of the study. Endpoints that happen more frequently or sooner give the trial a better chance to detect treatment effects within a set time and number of participants. This relationship guides how trials are designed and how quickly patients can learn if a new treatment is effective.